Hey, good morning, everyone. Let's get started. We have a very busy schedule today. Let's finish up lecture 7, and I would like to start on number 8 today as well. So, um, the last thing we need to do for lecture 7 is to talk about mutation. So, mutation is a permanent change in the DNA of a cell. Now, there are different kinds of mutation. There are large-scale mutation where a whole chunk of chromosome can break off and attach itself onto another chromosome. There are large-scale mutation where a section of chromosome pops out, flip itself, and then insert it elsewhere. We're not going to look at large-scale mutation. We are going to look at small-scale mutation, which are called point mutation. Which essentially happens at individual nucleotide level. There are two kinds of point mutation we will be talking about today. Base substitution, where we are replacing one nucleotide with another. If you just came in, the pink codon chart that I gave you before has a mistake. I only realized it yesterday when someone pointed out. Please pick up a blue one. Okay, it's on Rachel's desk. Okay, just go get one, and you can either throw this away or give it back to me. Doesn't matter. Do not use the pink one. Rachel, may you pass one back? Thank you. The other mutation point, the other kind of point mutation is insertion or deletion. Insertion or deletion, where nucleotides are added or removed from the DNA sequence. So leave some space here. We're gonna add more examples on it later, okay? We're gonna add more notes to it. Just leave some space. And then we're gonna come back to this. Now, I told you this before. Mutation will not be on quiz number 3, but it will be on quiz number 4. Okay, and you need to know how to do these kind of questions. Sorry, what was that? I don't remember. Quiz number 4, December 2nd, supposedly. Yeah, December 2nd. So, a couple weeks out. So leave some space. We're gonna come back to this. Angel, pick up the pink one has mistakes. Okay. All right. So it's your job now, okay? I'm not gonna keep on saying it. Whenever you see people sit down next to you, tell them to pick up a blue sheet, okay? All right. The mutation, for it to be a problem, has to occur at the DNA level. If you change the DNA, you are changing the instruction to make the protein. And when you do the transcription and translation, you're gonna end up having possibly a different protein. But we are learning it for the first time now. It's difficult to make the changes in DNA and then do transcription and translation. So to make it easier to understand, I am going to just do the mRNA first, okay? Keep in mind, the actual mutation has to be happening at the DNA level, okay? So point mutation, base substitution. Let's start with the first kind of base substitution. We have what's called wild type. Wild type just means normal. Okay? No mutation, normal. So let's say there is a mutation in DNA, and when you do transcription, it becomes UUU. And we are going to have a mutant. The mutant is the code that has the mutation. And in base substitution, I wrote it down for you. It's a change in one of the nucleotides, right? We're switching one nucleotide to another. So here I'm going to change it to UUC. Right? Now, what might cause this change? A number of things. It could be mistake that happens during DNA replication. If the polymerase 1 or 3 did not pick up the mistake, then it's going to persist in your genome. It could be caused by mutagens. Mutagens are chemicals or exposures that will cause your genetic information to change. Okay. There are many different mutagens in chemicals that we expose ourselves to. So please look up UUU on the brand new blue sheet, or you can look at the screen if you want, which is probably too small. Tell me what the amino acid is for UUU. Just yell it out when you have it. Yeah, so PHE, right? Yeah, that's an amino acid, phenylalanine. Then we have the mutation. We change it to UUC. What does UUC code for? UUC, yeah. Yeah, also PHE, right? So what happened? Well, nothing happened. Okay? In this case, changing the nucleotide does not result in a different amino acid. We call this a silent or neutral mutation. Is kind of like the best kind of mutation to have because it's not going to have any negative effect on you, right? Okay. So let me write down for you. Silent mutation, changing the nucleotide has no effect. There's no effects on the amino acid. As you know, there are 64 codons in total, but only 20 amino acids, which means there are redundancy. You have more than one code for each amino acid, right? For PHE, it's coded by UUU and UUC. For something like valine, it's coded by 4 different codons. Right? So this redundancy actually serves as a protective mechanism against mutation. Right? Even if you have a change in the nucleotide, it's not necessarily going to affect the protein that you're making. Right? And that's a good thing. Right? So that's neutral mutation. Remember, I told you a long time ago, between you and any other person in the room, how much of our DNA are identical? What percent? Do you remember? Yeah, 99.9, right? Okay. So the 0.1% difference between you and the person next to you is actually caused by these silent mutation. So we actually call these things SNPs, SNPs. Okay. Single nucleotide polymorphism. A polymorphism is a detectable difference in the DNA of two individuals. We will come back to talk about SNPs later on, bless you, in lecture 9. All right. But I just want to introduce you this term. Okay, that's silent mutation. Moving on, let's do another one. We have the wild type. Let's say this is U. You know what? Let's do something different. Let's say we have CUG. And I'm going to change it to CAG. Remember, base substitution means you have to change one of the nucleotides. It doesn't matter if it's the first one, the second one, or the third one. You can't change more than one, though, okay? Because that's no longer base substitution. Okay. What is CUG? Yeah, CUG is LEU. If you're still not sure how to use the codon chart, watch the video I post. And you absolutely must know how to use it for the quiz on Monday. Right? Otherwise, you're going to be losing a lot of marks. What about CAG? CAG? GL, right? Okay. Please double check that. So this is going to be a different amino acid. It's a mistake. That's why it's called a missense mutation. Do you think this is bad for your protein? Bad? Not bad? Yeah. Okay, here is a little secret. Whenever I ask you on a test or quiz or any time, if I ask you whether a mutation is bad or not, most of the time, the correct answer is it depends. It depends. All right. So the severity, or we can say the effects of the missense mutation depend on where it happens and in what protein it happens in. Here is an analogy to help you understand. If I say there is a long nail, okay, this long nail, and it's stuck in your car, is that going to affect the ability for you to drive your car? Yes or no? It depends. Okay, it depends. Okay, if it's in your tires, yes, then that's going to be a problem, right? Okay, you can drive a little bit, but eventually you're going to have a flat tire and it's dangerous to drive it, right? But if it's in your door, sure, it looks ugly and you're not happy, your car gets damaged, but you can still drive it, right? Okay, and that's the situation here. A missense mutation is not necessarily going to have a negative impact on the protein's function. It depends where the mutation occur in the protein. For example, some proteins, as you know by now, is going to be forming enzymes. An enzyme has a pocket, which we call active site. And so if the missense mutation happens in an amino acid that's forming the active site, then that could be a problem because it might change the shape of the active site and you will no longer be able to fit the substrate in it. You might no longer be able to carry the reaction, right? Then the enzyme will not be functioning. Okay, that would be an example of a loss of function mutation. Do you guys follow? All right. Now, that's the where it happens. But it also depends on what kind of protein is happening to. If this thing, like let's say it's not an enzyme, okay, let's say it's a protein now, and it has a loss of function mutation, it would be a problem if we are talking about a tumor suppressor gene, right? Like a red light. Loss of function in a red light can cause the cell cycle to accelerate, right? However, if we have a loss of function mutation in a green light, then maybe it's not as bad, right? If the green light stops working, that's not going to cause uncontrolled cell division, right? So not only does it depend on where in the protein it happens at, but it also depends on what kind of protein it is happening to. All right? So always think about all the scenarios. Are we together with the silent and the missense? There's one more. Nonsense. People love this one because everything we learn is nonsense. So let's start with the wild type again. Wild type. And let's say we have UAU. And I'm going to mutate it to UAA. Okay, please look it up. Okay. TYR? Everybody got TYR? Yeah. Tyrosine. And then UAA, you should recognize it as stop. Okay. So this causes the translation process to stop prematurely, creating a truncated. Truncated means shortened. I know, why can't they just say shortened protein? This causes the translation process to stop prematurely. Process to stop prematurely, creating a truncated or shortened protein. So let's say we have a mRNA sequence here. Take a look, please. AUG is our start codon. And then you're gonna have a bunch of codons, which I did not write. So normally you're gonna have the MET here, and then another amino acid. You're just gonna keep on building and building all the way until the stop, right? Okay, you would have like the MET here. Don't draw it, and then this would be the TYR. Now we have a mutation. This becomes UAA. So when the ribosome comes and try to build the protein, it will build MET and then amino acid, so on and so forth. And over here, as opposed to TRY, because it's a stop codon now, then it's actually going to stop here, and the rest of it is not going to be translated. So the protein, instead of this long, it's now this long. It's shortened, it's truncated. Is this going to have an effect? Yeah, right? You're missing half of the protein. That's a problem. Okay. Is this not this exactly particularly, but like, would you say nonsense mutation is generally worse than missense mutation? Would you say, here's my question. Would you say nonsense mutations are generally worse than missense mutation? What did I say?
Depends.
It depends. That's right. It depends. Okay. Depends on where it's happening. Right? I can have a missense mutation in the active site, and that would be worse than a nonsense mutation that's happening at the end, right? It doesn't have to happen in the middle. What if the nonsense mutation happens right here? Then maybe it's okay because you're losing just one amino acid, right? You're gonna stop anyways, but like you stop a little bit earlier, right? It's like your lab report, right? If you have a nonsense mutation in your lab report, you did the first page and the rest is gone. That's pretty bad, right? You're gonna lose a lot of marks, okay? But if you lose that last page, maybe it's okay, right? It's not detrimental, right? Do you understand? Right? So it depends, right? So you bet I'm gonna ask you that question on the
Okay.
on some assessment. We all good? Everybody happy? Yeah, don't look like it. Um, I'm gonna ask you to do this for me. All right. Let's say I have wild type. I gotta think about this. Okay. I need you to create a neutral, missense, and a nonsense. Okay, there are more than one possible answers, obviously, but go ahead and do it. Right on the other. Are you okay? No? Okay, so you write down the original version. Okay, what is UC what is it called? Okay, so for neutral, you need to change it so that it's still the same amino acid form. Okay. So we have to change one of the letters, one of them, so that the amino acid does not change. You know what it goes for the origin. So you have to look at UC where that has to operate. Right. Neutral mutations. What else is FDR? There's a bunch of them, right? So instead of using G, you can change it to one of these, and it's still yes. Right? You okay? No? Oh. Question? 1 second. Each one has been staying further away. Yeah. Yeah, so like usually if you want to do one, the last one. Yeah, and then for descent, you can change the first one or the second. But this is not right, because UH is actually stop. Right? So that would have been nonsense. Oh, just yes, I understand. Yeah, so like you have, yeah, I think this is why. It's up to you after your spiritual. The pink one has been fixed, so just move it over. You got it? So it doesn't change. Okay. You understand? That's right. So this is SER, right? You write SER. Yeah, so you need to change one of these letters so that the new one is also SER. Okay, but then you can only change one of the letters. So instead of UCG, these are all SER, right? You can actually change one neutral. Right, please change the G to C, right? And you put anything, right? You change it. This is still SER. Before it's SER, and after it's still SER. So there's no change. Like this one, but you have to change the same thing, you get a difference. But it's still make the same. Yeah, you got it. So this one. Yeah. So I write out what it is, right? And that's not correct. Because this is nonsense, they need it and stop. So this is not stop. Yeah. Right. So like just one of them. Okay. Now let me try to explain this one more time. You have to look up UCG first. Okay? UCG on the thing, okay? Hold on. So over here, UCG. UCG. That's SER, right? Okay, that's the original amino acid. Original amino acid. So I'm gonna write it here, SER. For neutral mutation, I need to find another code that's also going to be SER. All right. So if you look at here, you have a bunch of SER possibilities, right? So instead of UC, what did I say? UC what? Yeah, instead of UCG, you can do UCU. You can do UCC or UCA, right? Any of them is okay. So pick your favorite one. UCA. This is actually very difficult for me to mark because there's so many correct answers. Have some sympathy. Next, we have UCG, and we need a missense. Your best bet in creating a missense mutation is to change the first or second one. Okay? For neutral mutation is usually the last one. For missense, either the first or the second would be okay. So let's say I change the first one, right? Instead of UCG, change it to anything you like. ACG. Remember, you can only change one letter at a time. And you have to verify that you did it correctly. by looking up ACG. So if you look up ACG, it is THR. You switch the amino acid. It's a missense mutation. Okay? If you have questions, you need to ask. Okay, silent as a mutation. Last one. UCG. To become nonsense, you need to create what kind of codon? Stop codon. There are only 3 stop codons, right? UGA, UAA, UAG. So there's only one way to do this, and that is to change the middle one. That's it. All right. On quiz number 4, there will be a question like this exactly. I will give you a codon, and I'll ask you to create 3 different types of mutations. Please make sure you know how to do it. Okay. Quiz 4. Yeah, you can change. You can change first, second, or middle. Or sorry, first, second, or last. It depends on the situation, right? I'll give you another one. Let's say I have UAC. If I want this to be a nonsense, I cannot change the first one. I cannot change the second one. The only option to create a nonsense is to change the last one so that I get UAG or UAA. Okay. So to recap, for neutral mutation, usually it's the last one that you change. Okay. For missense, you can change the first or second, but double check your answer by looking up the amino acid. Okay. And for nonsense, it depends what the starting wild type is. Right? You might have to change the first, second, or the last, but never more than one. Got it? Okay. That was the easy stuff. Now, let's do insertion or deletion. But before that, I think I have some slides to go through with you. So here is more example for you to review on your own when you have time. Right? And it just shows you that the actual change happens in the DNA. Right? Once again, I'm doing the change in the RNA, so it's easier for you to understand. All right. Sickle cell anemia is a condition, it's a genetic condition where the gene that codes for hemoglobin is mutated. Hemoglobin is a protein that carries oxygen and carbon dioxide in your blood cells for you, and it has 4 polypeptides to it. It has a quaternary structure. We first learned about it in lecture 2. So when it's normal, then the red cell looks like this. And that's what your healthy red blood cell looks like. The hemoglobin protein, one of the chains anyways, is made up of, I think, 146 amino acids. And in the diagram here, the first couple ones are shown like that. The normal hemoglobin has this particular amino acid, glutamic acid, in this position, and it's a polar amino acid. And then there is a missense mutation that causes it to go from GAG to GTG. Remember, this is DNA, and it's the coding sequence in this case. So GTG, if you do the transcription, it's going to become like GUG, right? So if you look it up, it's going to be valent. The point is, the missense mutation here caused the amino acid to switch from a polar one to a non-polar one. And that switch is so extreme that it causes the hemoglobin to misfold and ultimately cause your red cells to become sickle-shaped or moon-shaped. People with sickle cell anemia, well, first of all, it's something that you're born with. So if you don't have it, then you're not going to like develop it later. But people who has sickle cell anemia, they are not able to circulate oxygen efficiently, so they are always tired. They have shortness of breath, and these sickle-shaped red cells actually get stuck in capillaries and cause quite a bit of pain. Okay, so there has been no cure for sickle cell anemia until recently. There have been a few successful gene therapy trials, okay, that has cured people of sickle cell anemia. We'll talk more about gene therapy later on. Okay, so just very briefly, a long time ago, we talk about proteins having different amino acids, 20 of them, and each of them has a different side chain, right? So we can actually group the side chains based on their properties, right? We have non-polar side chains. We have polar ones, positive and negative charge. So the missense mutation, if it switches from one nonpolar to another nonpolar, then again, maybe the effect is minimal. Okay, look at the structure. This is an H and this is a CH3. It's very similar, right? So may not be a problem. Right? But if you switch from a nonpolar to a polar, such as the sickle cell anemia situation, then it could be a big problem. Okay? So it's not just location, it's not just the type of protein. It also depends on what you are substituting. Right? Think about baking. I don't know if you guys bake, right? But if you run out of like cake flour, you can still use all-purpose flour. Your thing's gonna turn out fine. Okay. But if you substitute the sugar with salt, then it's not gonna be okay, right? Okay. So not all substitutions are equal, in other words. Okay. It depends. All right. So to understand frame shift, to understand insertion or deletion mutation, we have to understand something called reading frame. What's going on here? Discard. Cancel. Okay. So insertion deletion means you are inserting extra nucleotides into the sequence, or you're deleting nucleotides from the sequence. And depending on how many you insert or how many you delete, the outcome is going to be different. Okay. Sometimes you cause what's called frame shift mutations, other times not. So let's try to understand this frame shift mutation. Our ribosomes can only read nucleotides in triplets, right? Because every 3 nucleotides represent a codon. So let's, for example, pretend we can only read sentences, words in 3 letters interval. So the first sentence here, right? All the words are 3 letters, and we can read it just fine. The big cat saw the fat dog. Now let's take a look at the next sentence. Suppose I take out the B, then what does the sentence become? The is still fine, but without the B, remember we can only read in triplets. So the next word that forms will be IGC, right? Which I don't know what it means. And then everything else is actually shifted after that, right? So this is what we call a frame shift. A frame shift. You shift the reading frame and nothing makes sense anymore. However, if we delete 3 letters at the same time, so if we delete the word big here, the sentence structure is maintained. Okay? The cat saw the fat dog. You can still read it, right? It's still pretty much same as the original, but you are missing like an adjective here, right? You're missing one of the words in the sentence, right? But the frame is retained. What is the problem here compared to the original? Do you notice the problem? Spot the difference. What's that? No, I don't think anything switched places. Extra what? There's an extra letter here, right?
Yeah.
You see that? Yeah. So if you put an extra K, and now it's like THK, and then again, everything shift. right? Doesn't make sense anymore. Frame shift. What's the problem with the last one here?
Red.
Yeah, the word red, right? But it's 3 letters, right? So it's fine. It's fine. You maintain the sentence structure. You just have an extra word. No frame shift. Okay? So back to our notes. For insertion or deletion, sometimes they're called indels, we have 2 situations. Multiples of 3. If you add or delete multiples of 3 nucleotides, tell me what's going to happen. If you insert 3 nucleotides, what's going to happen to your protein in the end? If you insert 3 nucleotides. Oh dear. If we insert 3 nucleotides, like if we insert U into the sequence, what's going to happen to your protein? Okay, it doesn't stop, yeah, but more importantly, it's going to have a extra what? Amino acid, that's right. So if you insert or delete multiples of 3 nucleotides, you're going to end up having extra or missing amino acids, right? Stay with me, stop copying. If I delete 12 nucleotides, what's going to happen to your amino acid sequence? Are you going to have extra or are you going to have missing ones? Missing. How many are you going to be missing? 4, right? 12 divided by 3 is 4. Okay. Why do I divide by 3? That's right. 3 nucleotides for 1 codon, right? Understand? Good. Not, yeah, keep copying. No, not multiples of 3. Then we're going to have frame shift mutation. And we're going to look at the consequences of frame shift mutation in a bit. Hands up if you have the study guide with you. Okay, good. If you don't have it, I guess you can look it off the screen, or you can look at it on your computer. This is what question is this? For those who have it, I don't remember it. 11, thank you. Right, so there will be something like this on quiz number 4 as well. So this is the template. It actually says in the question, I just did not copy it. Yeah. And over here, this is the wild type. Right, so what I need you to do, and work together, please, is to chop them up into triplets. I need you to do the transcription and translation. And we're going to move on from there, okay? When you have the sequence, let me know. I'll come over and check it. Oh, darn, I did not print the answer. That's okay. Don't call me over then. Just do it first. This is template DNA. If you want to go from template to messenger RNA, you have to do the base pair. Set them up in template. Template DNA. So if you want to go from template to mRNA, what's the relationship? You copy it, or do you have to do base pairing? That's the base pairing. What the B pair with B pairs with A, B pairs with B pairs with A, and the base pair. This pair. Right here. Probably down to G, remember? I will. B pairs with G. Oh. So over here, we have AUG, right? Please check my work. You have to use the complementary base pairing rule, right? And then you look them up. Can someone confirm that, please? CGC is wrong? Yeah, it is. Thank you. It is ARG, right? It's okay now? All right, this is the wild type, huh? That means it's the normal one. Now we're going to have a mutation. All right, so there's the mutation. It's a good idea to chop them up into triplets. Compare the mutant with the wild type. See if you can find the mistake. You have it? Mistake? It's hard for you to tell me, right? I guess. Where is the mistake? Which codon? So if you compare it letter by letter, TAC, TAC, ACC, ATC, the mistake is right here, right? Okay. This is a base substitution. You're switching out the C with the T, and it's happening in the DNA level. So now we're going to go ahead and do the transcription. So TAC will be AUG. It's no longer lined up, but that's okay. ATC will become UAG. And the rest would be the same. Okay. This is the mistake. This is the mutation. Okay, come here. The pink one is wrong. Okay, throw that away. All right, what is AUG? AUG is the start codon, so it's MET, right? And then UAG is the stop codon. Then you don't have to continue. Okay. Once you stop, you stop. Before we talk about the effects, let's talk about the kind of mutation. It's a base substitution. What kind of base substitution? Nonsense, right? So I need you to be specific on the test quiz. Right? Don't just say nonsense. Don't just say base substitution. Say both to get the full mark. Will there likely be effects? Well, yeah. I mean, in general, it depends. But then for this specific example, is there going to be a problem? Yes, because the protein is gone. You didn't make the protein. You only have the methionine and then it stopped. Okay? So will there likely be effect? Yes. The entire protein is missing. This would be loss of function mutation. You lost the protein. It's not going to function. You're not making the red light. That could be problematic. Right? If this was insulin, you're not making insulin anymore. Okay? In fact, hemoglobin, there is a type of mutation that's nonsense, and it caused a whole chunk of it to be missing. People with this kind of mutation has a condition called thalassemia, which you might or might not have heard of, but it's one of the things that they screen for in the mother when they are pregnant. Okay, because if the baby is born with thalassemia, they are going to have severe anemia. Okay, and they have to have blood transfusion regularly to survive. All right, let's take a look at the next one. The next one. What's the first thing you do when you get a question like this? You chop them up, that's right, into triplets. Now watch. If you chop them up into triplet and you end up seeing like a leftover, that's a sign that this is not a base substitution. Okay? What is it though? Insertion, right? So your job is to identify the extra nucleotide, right? So go ahead. Where is it? So if you found it, go ahead, circle it, and then finish the question.
Well, how do I know it's Roman? Because the first, the very top one, that says WT means wild type. That's the origin. So that's the one you compare with. Yeah, so if you compare the first one, then TAC, and then it's also TAC, right? Because of mutant. Number two is an ACC. Yeah, daughter.
It's like neutral.
Oh, no, it's always, I would always give you the preference. Yeah, we do this one, right? So you can compare letter to letter, okay, and then see where the problem is. Okay. Yeah. So TAC, right? And then here it says ACC, and here it says. So you see that's the problem, right? Okay, that's the problem. You identify the division? So this is the number two windows. Okay, where's the problem? Wow, okay. But it's maybe copy this. No, because there's an extra letter, right? So something like this. Are you okay? Find a mistake? No. So just one letter by then. B A C and then G, right? That's the extra one. See, everything else is the same. Watch. ACC, right? You see that? And then TTG, right? Everything else is actually the same. Compare this with this, you see? Yeah. But the extra.
Okay, so again, in case you didn't catch what I said, when you chop it up into triplets and there is one left over, it suggests that you are looking at an insertion mutation. So you have to find the extra, let me get this out of the way, the extra nucleotide. Right. So over here, it's TAC. Oh, hold on, what did I do? Okay, so here you have TAC up here, and it's also TAC. Then ACC in the original, but now it's GAC. Now, you might be tempted to say it's a base substitution or something like that, right? That the A get changed to G, but then that wouldn't make sense because A gets changed to G and then C gets changed to A, then that's like more than one changes, right? Okay, and I told you if there is a leftover, then it's not likely to be a base substitution. It's an insertion. So what actually happens is you've inserted an extra G in it, okay? Extra G. So now, if we do the transcription, you just, follow the base pairing rule. Do you guys have any question for me? Okay, so I'm just gonna do the translation for you. Okay, again, please check my work. Okay. So let's compare the wild type with our mutant. It's met, which is good. And then everything else is wrong, right? Everything else is wrong. And in fact, we're supposed to stop there, but we missed the stop because of the frame shift. So this is going to go on for some time until maybe it hits another stop later. What kind of mutation is this? What's that? Frame shift, yeah, but before that, what caused the frame shift? Insertion, which resulted in frame shift. And the frame shift actually resulted in extensive missense mutations. Okay. Everything is a mistake. All of them are missense. Will there likely be effects? Absolutely. The wrong protein is produced. Right? It's like changing the recipe completely. You wanted to bake a cake, you end up making pot roast. Totally different. Because all the amino acids are different. Make sense? Okay. I don't even know if this protein is going to be functional at all, right? Or like what it does. All right. I'm gonna have to take away your break today. I'm very sorry because we have a lot of things to do. I mean, if you need to have a break, then have a break. I'm not going to stop you, okay? But I'm just going to keep talking. We're going to do one more example here, and then we're going to move on. Okay. And again, if you need to walk around, please do so.
Sorry, could you just bring one over there? Yes.
About yes, but the protein. Did it change? Yeah, I mean, let's see, but did the protein change? No, so what gonna be the change? Protein is fine. Okay, MVP, PRP, and ARP. So even you change the DNA, but the protein did not change. That's why it's going to keep neutral, because the neutral. Neutral? Good. Okay, so here, again, you want to chop them up. And the mutation is right here, right? Okay, so I hope you found that. Everything else is going to be the same. I'm not going to repeat it. You should when you do it. But the TTA is going to be AAU, right? And if you look up AAU, it is ASN. So the third one is actually same as the original. This is a situation where you change the DNA, but it has no effect on the protein. And so, no, there will be no effect. And this is a base substitution that resulted in neutral mutation, neutral or silent. Is that okay? All right. The next thing we're going to learn is difficult. So, I mean, that's what I said about everything, right? But it's actually true. Okay? This one is actually difficult. So please stay with me and ask a question. I think I lost my mouse. Where is it? Okay, we have 4 more examples to go. And there will be questions like these on the quiz number 4. So we've learned about the different types of mutations. We learned how to identify mutations. The last thing we're going to do for lecture 7 is to create mutations of a certain kind. This is from your PowerPoint slide. Create a base substitution. By the way, there are more practice in the study guide, right? So help yourself with it. Create a base substitution in the coding strand below that will result in a missense mutation. So this is coding. This is coding. And I want a missense mutation. So there are multiple ways to do this. Right? And you know, as long as it's correct, it's fine. Like you can choose how you want to approach this. So, for example, let's take a look at this one here. This is CAG.
and this is coding. So what would the mRNA be? Do I copy or do I do base pairing? Copy, right? If it's coding, you copy. So if it's CAG here, it is still CAG. Of course, you change the T to U, right? But there are no T in this particular one, so that's fine. I'm not doing the whole strand. I'm just doing one of them. What does CAG code for? GLN. So I want a base substitution, meaning I change one of the nucleotides, and I want it to be a missense mutation. If you want to create a missense mutation, what's your best bet? First, middle, or last?
Last.
First or second, right? Yeah, first or second. So pick your favorite letter. Cross out the C, change it to whatever you want. I'm going to change it to A. If you change it to A, this is going to be A as well. And it's no longer GLN. It is going to be what is it going to be? LYS, right? That's a missense mutation. That's not the only way to do it. I'm going to try another one right here. You only have to do it once, okay? You don't have to do so many. I'm just doing another example for you, okay? You could have picked this guy. And this would have been AAU when it's being transcribed. If you look up AAU, it is ASN. So you can change the middle one. You can change it to C or something. And then now you look up ACU. ACU, which is THR. So that's another example. And remember, this whole thing is being recorded. If you need to watch it again to understand more, you are encouraged to do so. Are we good with this one? Okay. So on the quiz or test, just pick one and then do it like this. You don't have to transcribe the whole thing. You don't have to translate the whole thing. Right? In fact, it would be a waste of time to do so. Give this a try. I'll take it up in a couple minutes. Oh, don't forget to hand in your lab before you go, okay? If you have it, maybe you can raise your hand. I'll come get it. Anyone else? No. Does anybody want to share their answer? Any brave soul? Yeah. Go for it.
So I changed the CAG, the second one,
Uh-huh.
to GUC, which is the
To what? I'm sorry.
GUC, right?
Okay.
And then, which is valve, but then I changed the middle one to GCC, which is
Okay. Yeah. So
ALA.
what you're doing is you're creating a sense mutation, but the question asks for nonsense mutation. Okay. So that's yeah. Not necessarily. Okay. So let's see. A nonsense mutation means you have to create what kind of codon? Stop. Please recite the stop codons for me. Okay, I think I'm hearing UJ, UAA, and UAG. I think that's what you guys said, right? Now that's the mRNA, okay? Listen carefully. It will save you some time on the test. This is the coding strand. So can you change these stop codons into coding codon for me? What would it look like on the DNA? What would UGA look like on the coding DNA? What is it? It's TGA, isn't it? What's the relationship between coding and mRNA? They are the same, right? They're the same, except the T and the U switches, right? So that's going to be TGA. What about UAA? What would it be? TAA, right? Okay. And the last one, please. Since this is the coding strand that's given, all you have to do is change a letter to create one of these. So over here, I see CAG. If I change the G to a T, then it's a stop codon now. Okay. If you do transcription of TAG, it's going to become UAG, which is a stop code. Okay. Is there another possible answer? Which one? AAG. And how can you change AAG? The TAG, right? So both answers are correct. I want to emphasize on a test, you only do one of these, right? Not both. Okay. So stop codon is a little bit tricky. Nonsense mutation is a little bit tricky because it's not going to work for all of the codons, right? Like this one is not possible to change to a stop codon with base substitution. You cannot just change one nucleotide to make it into a stop codon. So it's not always going to work. All right. Question or? Yeah, okay. So the last one is TAA, which is already a stop codon, right? So we're not going to change the last one because it's stop. Even if you change it and it becomes stop again, then actually that's not going to have any effect, right? Because you would have stopped there anyways. Are we together? Okay. Don't wait until the fourth quiz to study these things, okay? Right. Study for quiz 3 and then also start studying for this stuff. All right. Because I told you, statistically speaking, quiz 4 is the worst performing quiz in the course. Okay. Because there's a lot of problem solving. I will try to help you with more review, but you know, Just a warning. This one. Create a base substitution in the template strand. Template strand. And I also want nonsense. Okay. So we will once again start with the stop codon. Okay. You got A. You are awesome. You are great. And then what would it be as a template code? What would UGA look like on the template DNA? Yeah, I heard it. Just louder. ACT, right? Because the relationship between template and mRNA is that they are, what's the word I'm looking for? Complementary. Complementary. This one is ATT, ATC. Okay, so all you have to do is find where you can change it to one of these things. Yeah, you can change the TTC to ATC. Alternatively, you can change AAT to ATT or ACT. The last example. Create an insertion mutation in the coding strand below that will result in a truncated protein. And so I'm just going to copy this stuff here from above. This is coding strand again. And how do we create a truncated protein? What do we have to introduce to make it shorter? Stop, right? We have to introduce a stop. Right? But this time, the question wants you to create a stop codon, not with base substitution, but with insertion. Okay? So you need to insert something to create one of these stop codon. So for example, we can do it right here. We can put a T there. Okay? So your ribosome will read it as AUG, GAA. This is coding, right? UUC, and then the next thing it will read after the insertion would be UAA, which would be a stop. Okay. Alternatively, I can insert it right here. So when the ribosome reads the code, it will be AUG, and then what's the next triplet? AUG, and then the next one would be UGA, right? And that would be a stop codon as well. All right. I'm going to show you a mistake that people make. Okay, don't erase yours, obviously. Right? Sometimes people do this. They put a T here because they think you will get TAA, right? You see, TAA is one of the subcodon, right? But that's not going to work because when the ribosome reads it, it's going to read it as AUG, and then GUA. It's not going to skip the G and then read the TAA, right? So you have to make sure whatever you insert, it's going to be in the correct frame to create the stop code. Okay? All right, very heavy stuff. Lots of things for you to study from. I love to let you go home, but we actually have to do a little bit of 8 as well, okay? Because Monday, we're going to have the quiz, and after the quiz, people usually have a hard time focusing. So let's get some of this stuff out of the way first, and then we will continue on Monday. So as I told you, it's going to get progressively harder. 8 is harder than 7, and 9 is the hardest one. All right. And then 10 is okay. 10 is easy, right? And then we'll be done the course. So next semester, even harder because it's 6 hours per week as opposed to just 4 hours, right? So I don't know. Maybe we'll meet again. Maybe not. I don't know. We'll see. So number 8 is heredity. Okay. Heredity is about passing the genetic material to the next generation. And, even though it's difficult, it's actually a fun lecture, right? We'll talk about things like, if you have blue eyes, your spouse have brown eyes, what % of your kids are going to have blue eyes, right? The basis of heredity relies on the process of meiosis, right? Some organisms, as you know, reproduce asexually. That's just through mitosis, right? One cell divides and then you have 2, like plants, like the starfish that I showed you. But we as humans, we reproduce sexually. That means we need to produce sperm in the male and eggs in the females. And then when the 2 comes together, you create the offspring. So the purpose of meiosis is to create reproductive cells. And the process of meiosis actually causes your genetic material to be reduced by half. Right? How many chromosomes do you have again? 46. Right? So when the female makes the egg, you take 46 and you divide it by 2, the egg's going to have 23. Okay? And then for the male, the sperm is going to have 23. When the sperm and the egg fertilizes, the sperm fertilizes the egg, then 23 plus 23 is going to give you back the 46, right? Okay. So meiosis is the basis of sexual reproduction. There are a few new terms here for you to learn. Humans are diploid organisms, which means we have 2 sets of chromosomes. Okay? We know that already. 1 set from mom, 1 set from dad, right? So all our somatic cells basically means bodily cells, body cells. Right? Your liver cells, your brain cells, your kidney cells, what have you, they are all diploid. They have 2 sets of chromosomes. So we say 2N, okay, 2N, where N is 23. 1 set has 23 chromosomes. We have 2 sets. 2 times 23 is 46, right? The gametes. Gametes is a fancy word for sperm and egg. Okay? The gametes are haploid. They only have 1 set of chromosomes. Okay? So sperm and egg, it's only N as opposed to 2N. So the sperm is N, the egg is N. When you put them together, N plus N gives you 2N, right? Diploid is not the only possibility in nature, okay? it's possible for things to have 3 sets of chromosomes, which we would call triploid. And so in this example, N equals to 3. One set has 3 chromosomes, right? So 2N would have 6 chromosomes, right? 2 of each. 3N means you have 3 of each. So a total of 9 in this case. You can have tetraploid, 4 sets. And more than that, it's called polyploidy. Strawberries have 8 sets. Yam or sweet potato, they have 6 sets. Okay. So it's very common for plants actually to have multiple sets of chromosomes. Okay. Two sets. Yeah, it depends on what the N number is, right? Okay, so in this particular example, N is 3. But if N is like 23 like us, something that's triploid would be 23 times 3, right? Which, sorry, one more time. No, they're not stuck together. So we count them separately. So that's 1, 2, 3, 4, 5, 6. Okay. Now, one of the questions that bothered me earlier on was how did they make seedless watermelon? Okay. So to have a plant, you need to have a seed, right? So if the watermelon has no seed, how are you going to grow the seedless watermelon, right? So it turns out regular watermelons are diploid, so 2 ends. So there is a special breeding technique that forces the watermelon to become 4 ends. Okay, 4 sets of chromosomes. Okay. And then what they did is they take the 4N watermelon and then they breed it with the 2N watermelon. And in the end, it creates a 3N watermelon. Now, you're thinking, how does 2 and 4 becomes 3? All right. So in sexual reproduction, when you produce the sperm and the egg, you divide the number of chromosomes by half, right? Okay. Plants also have sperm and eggs. We just don't call them sperm. Okay, we call them like pollens. Okay, pollens and eggs. Okay. So here, for the 2N watermelon, their gametes are going to be N, right? 2N divided by 2 is N. You with me? So like the quote unquote sperm would be N. What about the eggs? How many N in the eggs? 2N, right? So if you take the N and the 2N and you put it together, you end up having 3N. Okay, here is the takeaway important message. Any organisms that has odd number set of chromosomes cannot have sexual reproductions. Okay? Organisms with odd number set of chromosomes cannot reproduce sexually. Why is that? That's because if you have odd numbers like 3N and you try to make sperm and egg, you will have to divide this by 2, right? But you can't. Right? 3 divided by 2 is 1.5. You can't have 1.5 sets of a chromosome in the egg and 1.5 sets in the egg, right? And in the sperm, right? You're going to be missing genetic information. So anything that has odd number of set of chromosomes cannot produce gametes, and therefore they have no seeds in the case of watermelon, right?
Is it like, for example, like a ginkgo tree and of a ginkgo? And some of them don't produce any ginkgos on them.
I have no idea what that plant is, but we can look it up later on. Yeah, I've never heard of it. It's a what? It's in grocery stores? Okay, I see. Oh, wow.
So for example, like an oak tree, right?
We'll get back to that. Let's finish this first, okay? All right, so sexual reproduction. A lot of new words for you again, okay? The process of sexual reproduction begins with meiosis in germ cells found in gonads. Okay, so a lot of G words here. Right. So gametes are, what are gametes again? Do you remember? Sperm and egg. Okay. How do you make gametes? You have to take germ cells. Okay. Germ cells are cells that will become sperm and egg. Okay. All the cells in your body are somatic cells, but the ones that will become sperm and egg, we call them germ cells. Okay. They are not sperm and egg yet. They will become sperm and egg. Where do you find the germ cells? You will find them in gonads, which is a fancy word for reproductive organ. Reproductive organ. What's the male gonad? Where are sperm made in males? We have 3 males in the class. You guys know where the sperms are made? Okay, it's the testicle, okay? It's okay to say the word. And in female, where are the eggs made? Ovaries, there you go, okay? So the germ cells in the ovaries are going to become eggs. Okay, we're going to see how that happens later on. And in the male, the germ cells are found in the testicles and they will become the sperm. All right? Wonderful. So to put it all in a sentence, germ cells are diploid reproductive cells. They have 2 sets of chromosomes and they will give rise to haploid gametes. Right? You cut the number of chromosomes in half by performing meiosis. Fertilization. A zygote is formed when a sperm fertilizes an egg. Right? So as soon as the sperm gets to the egg, it will infuse its genetic material, and then you have a fertilized egg that has a full set of genetic information. That single cell that is being formed is called a zygote. And we have trillions of cells in the body, but we all began with one single cell, the zygote. Okay? And then it's going to start to divide and divide and divide, and eventually the zygote will become the embryo, right, which then becomes the baby. So the process of meiosis is going to involve 2 subsequent division. Right? You split it once, and then you split it again, and that's how you have the genetic information. Oh, sorry. I'll try again. So for meiosis, we're gonna have to take a note on that. Let's start on the note, okay? And then we'll finish up later. So sorry, guys. Okay. So meiosis is separated into meiosis 1 and meiosis 2. What are the 4 phases of mitosis? Do you remember? Mm-hmm. Yeah, keep going. Good. So in meiosis, it's the same thing. You will have prophase, metaphase, anaphase, telophase 1, and then you will have prophase, metaphase, anaphase, telophase 2. Okay, it just happens twice. Right? As always, I'm not going to ask you to describe the meiosis process. I'm not going to ask you to draw it out. but you will have to know the difference between mitosis and meiosis. And that's something I will summarize for you later on. Okay, so let's start with prophase number 1. Okay, so for prophase number 1, okay, I'm just going to draw 2 chromosomes here. Okay, they are already duplicated, and that's where they're stuck together. And then over here, please use color, okay? Otherwise, it's not going to make sense. There's no. All right. So some terminologies to refresh your memory. Or maybe you can help me because you need to know it for your quiz anyways. This red one and this red one, they are stuck together. What do we call them? The whole thing is a chromosome, yes, but specifically this red one and this red one. Sister what? Yeah, TID, right? Sister chromatids. Sister chromatids are identical to each other in every single way. Okay? You just copy the whole DNA. How about the red one and the blue one? What do we call the red one and the blue one? Okay, that's fine. Make sure you know it for the quiz though, okay? Homologous chromosomes. Homologous chromosomes are chromosomes that are same length, same size, and contains same type of genetic information. Okay. So what that means is this. You don't necessarily have to copy this part down. Okay. But let's say I have chromosome 1 here from mom, and then we have chromosome 1 here from dad, right? The genetic information would be like over here. That's like capital A, and then maybe also capital A. Over here, brown hair, brown eyes, blue eyes, right? Okay, curly hair, straight hair, no dimple, right? Earlobe is attached, earlobe is detached. Right? So basically, you have the same set of genetic information in the same order. However, the version might be a little bit different, right? You might have brown eyes from one parent and blue eyes from the other. You might have attached earlobes and detached earlobes, but they might both not have dimples. So homologous chromosome. You need to know these terms. Remember, on the quiz for Monday, there will be fill in the blanks. Okay? And I will not be giving you words to choose from. Please practice those from the study book. All right. So in prophase 1, the homologous chromosomes will actually find each other. The chromosome 1 from dad will find chromosome 1 from mom, and then they will pair up together. Okay? 2 will pair up with 2, 3 will pair up with 3, and so on and so forth. Okay? And in the end, they form this whole thing. Okay? This is called a tetrad. Tetrad means 4. Okay? 4 sister chromatids. 1, 2, 3, 4. Okay, so let me write it out for you, and then we'll be done. Homologous chromosomes find each other to form a tetrad. there will be exchange of genetic information between the homologous chromosome. So how does it look? And let me explain first before you talk more. Right, so you understand these are homologous chromosomes, right? They will find each other, they will form a tetrad, and then they will exchange genetic information. So for example, over here, originally the mother one, the red one, is capital A, capital B, and then small c, small d, and then capital E. But then what happens is they're going to exchange a little bit of genetic information. So maybe switch up the E's here. Okay, maybe switch up the D's, although switching up the D is not going to do anything since it's the same thing anyways, right? Okay. But the point is to shuffle the genes on the mother chromosome and the father chromosome so that in the end you get something completely different from the mother and the father one. Okay? And that's important because our kids don't look identical to us, right? It's because of this shuffling. If you don't shuffle the gene, you will end up either passing on the exact same red one that you got from your mother or passing on the exact same blue one that you got from your father, right? Then there will be very little genetic variation in the human population. Okay? So over here, they're gonna, cross each other. It's called crossing over. And afterwards, this is, of course, oversimplification of the drawing, but afterwards it's gonna look like this. Let me try to do this better. We are done after this, I promise. Okay, you're gonna have a bit of blue on the red. And then you're gonna have a little bit of red on the blue. Okay. Do you understand? Now, in reality, it's not just the end that gets switched. Okay. In reality, it's more like blue, red all over for both of them. Everything gets shuffled. Okay. And I know in the diagram I only do the shuffling for one of them, but it's actually happening to both of the chromosomes as well. Okay. One last question. Do you think there will be crossing over between this one and this one? The red and the red? No, right? Because there is no point. The red and the red, remember, they're sister chromatids, and sister chromatids are identical to each other. So even if you switch them, you're just going to end up having the same thing. It's not going to create genetic variation. Okay? Thank you so much for your attention. Study hard for the quiz, and I will see you all on Monday.
You were talking about